Hello and welcome, folks. I am Mr. Hamele, your teacher for AP Biology this year, and these are the video notes for those who weren't able to see the live notes in class. So this is the first part of our Unit 3 notes. We'll call it 3A, dealing with cellular energetics. In this section of the notes, we covered over a number of days, but we're going to cover 3.0 and 3.4, which is basically about energy. What is it? In the universe and in living things. Then we'll take a look at enzymes, specifically looking at enzyme structure and enzyme catalysis, or how enzymes speed up chemical reactions. And the last part of these notes, we'll be looking at how the environment that surrounds an enzyme can impact its function. And on here you can see the page numbers in our book, our current book in Campbell's Biology, that deal with these subjects. So let us begin. Part 1, 3.4 cellular energy. All living things need energy, guys. So first thing I want to take a look at is just kind of the concept of energy in general and thermodynamics. The energy of life. The living cell is a miniature chemical factory where thousands of reactions occur. All right, that's what life is. And the cell extracts energy stored in sugars and other fuels and applies that energy to perform work. So we take the energy out of the food we eat. The body's got some mechanisms that we're going to talk about in this unit to take that energy stored in the food and transfer it into things like moving our arms and breathing and things of that nature. Some organisms even convert energy to light, as in bioluminescence. Here's a cool picture. I have never gotten to see this in person, but I would love to sometime. Looks like a pretty cool phenomenon. All right, so let's just recap on the concept of metabolism. An organism's metabolism transforms matter and energy, subject to the laws of thermodynamics. Basically, metabolism is all of an organism's chemical reactions, everything that goes into that's happening inside the body. It is an emergent property of life in that it arises from the orderly interaction between molecules. So life makes this happen. And an important concept for us is the metabolic pathway, where you go from a specific starting molecule to a specific ending molecule. That usually doesn't happen in just one single step, one single reaction. Usually there's a lot of reactions along the way, and those reactions are often catalyzed by enzymes. So here in this diagram, it kind of shows you that cascade of events that happens in most chemical reactions. You don't just go from point A to point D in this case. There's a bunch of intermediary molecules, and the reaction from A to B and B to C and C to D are all catalyzed by different enzymes, and that is pretty typical in living systems. So the study of how energy flows through living organisms is known as bioenergetics. And two big things here I want you to understand is the difference between catabolic and anabolic pathways. Catabolic pathways release energy by breaking down complex molecules into simpler molecules. Okay, so if you start with a larger molecule like glucose, and you can break it down into smaller molecules, in that process, you'll generally release energy. Prime example of that is cellular respiration, where we take glucose, break it down, and in the presence of oxygen in aerobic respiration, and we produce all the energy that we're using, or most of it. Anabolic pathways is kind of the reverse of that. It's where you have to consume energy to take these smaller molecules and build them up into something bigger and more complex. Prime example of that is photosynthesis, or making proteins from amino acids. Okay, we take some outside energy, we use that to bind these together into more complex molecules. So catabolic, big, break it down to release energy. Anabolic, we start with small things, put them together, but that takes energy. Just a little review. You probably know all these terms already, but want to make sure that we're all clear on some of these different forms of energy that do work. Kinetic energy is just the energy of motion. And heat or thermal energy is the kinetic energy we associate with random movement of atoms or molecules. We measure this with temperature. Potential energy has the potential to do work. It's not releasing energy at that point in time, but it could. And then we have chemical energy, which is basically stored, potential energy stored in chemical reactions. Okay. Big point here at the bottom, energy can be converted from one form to another, but that's not 100% efficient. Okay. Every time you change energy from one form into another form, we're going to lose some to heat or some random motion of some other molecules. We're not going to get 100% transference. There's always going to be some energy loss. Not destroyed, lost, goes to something where we can't use it. And that brings us to the laws of thermodynamics. According to the first law of thermodynamics, the energy in the universe is constant. There is this much energy in the universe, and there has always been, and there will always be. And it says we can move that energy around within the universe, but we can't destroy it or create it. We can't get more, and we can't wind up with less. Okay? It's also known as the principle of conservation of energy, which you may have heard of before. This bear there is kind of exemplifying that. There's chemical energy stored in that fish he's eating. And after he eats it, it's still going to be there. It's just going to be in different forms. The second law of thermodynamics states that during every energy transfer or transformation, some energy is going to be unusable and is often lost as heat. Mentioned that before. Every energy transfer or transformation increases the entropy or the disorder of the universe. So entropy means things fall apart. I don't know if anybody's ever read the book by Chinua Achebe. It's a really good book if you haven't. But things tend to fall apart. They go from more complex to simpler, more bonds to fewer bonds. And as you go from more complex to simpler, you're going to release energy, which is going to get lost to other things, heat and things of that nature. Eventually, according to this theory, the whole universe will go down to its simplest forms and be completely spread out. Living cells unavoidably convert organized forms of energy, like we've used glucose a lot, we'll keep using that example, into energy like heat. Okay. Spontaneous processes, spontaneous chemical processes can occur without energy input. They'll just go, things will fall apart. These molecules will spontaneously on their own. Eventually, the bonds will break and they'll fall apart and will go towards entropy, right? Now, that can happen very quickly if it's a bond that's easily broken with just a little bit of heat input, or it could take thousands of years for it to break apart, but it still can happen spontaneously. So for a process to occur without energy input or spontaneously, it must increase the entropy of the universe, which means it's basically going to be creating more disorder. So more complex to less complex. Okay. Cells create ordered structures from less ordered material. That's a key defining characteristic of life, right? We, living things, create order within our cells. Okay. Organisms also replace ordered forms of matter and energy with less ordered forms. You can see that bear took that nicely organized fish and turned it into moving carbon dioxide, moving water molecules, splashing water, air movement, heat release. So we took that organized and we turned it into disorganized. So overall, energy flows into an ecosystem in the form of light and exits in the form of heat. We can't break the second law of thermodynamics. The universe is always tending towards disorder or entropy. Even living things that fight against that for a time, in the end, we create more disorder than order. And this bottom one is why energy flows in an ecosystem in the form of light. That's why we always need an energy input from somewhere on this planet to keep life going. Okay. The energy is not 100% transferred. If it was, how we got matter cycles like the carbon cycle. All the molecules on the planet keep getting recycled over and over again, which is great. But energy, we need a constant input because we're constantly losing it, mostly as heat. So we need that constant input to keep things moving. Just said that on the last slide already, so I don't need to repeat myself. All right, so let's take a look at the concept of free energy, or sometimes known as Gibbs free energy. And nothing's really free, guys. The free energy change of a reaction tells us whether or not the reaction occurs spontaneously. All right, so what does that mean? We want to know, as biologists, which reactions occur spontaneously, that'll happen on their own, and which require the input of energy, because that determines why do we need energy. We need to take in energy so we can do those reactions that we need to stay alive that take energy. So we need to determine the energy changes that occur in different chemical reactions. So based on the second law of thermodynamics, will a reaction that occurs spontaneously release energy or need energy input? Yeah, it's going to release energy. If it occurs spontaneously, it can release energy. If we need energy input, it's not going to occur spontaneously. We're going to have to give it some energy to make it go. And that's why living things need to eat or photosynthesize or whatever it is to get our energy. We can measure that with a free energy change. Delta there, the little triangle, means change. G is free energy. A living system's free energy is energy that can do work when temperature and pressure are uniform as in a living cell. You do not need to know how to calculate Gibbs free energy for the AP Bio exam, so we're not going to get into that. But you should know the concept of energy and know the difference between negative and positive change in G or free energy. Only processes with a negative change in G are spontaneous, meaning they give off energy. Okay, negative means you're giving off energy. A positive change in G means you needed to add energy to get that reaction to go, to get those things to bind. Okay, whereas if the bonds are being broken, generally you're going to release energy. You're going to have a negative G. Okay, so they increase entropy. And if we can harness energy in those spontaneous processes, we can do work with that. And that's what living systems are all about. So, free energy is a measure of a system's instability or its tendency to change to a more stable state. I'll put something up here on the screen. When I was doing this live, I kind of stood up nice and tall, like if I was blocking in football, stand nice and tall and my feet together, nice and tall, but I'm relatively unstable, right? If somebody knocks me over, I'm going to fall and hit my head hard. And then I squatted down into a good blocking stance, wide feet, lower center of gravity. I was more stable. It would be harder to knock me down. That's kind of a way to think about free energy. Higher energy, less stable, more organized. Okay. If you can drop that energy, lose some of that energy, and drop to a simpler, lower energy state, okay, you're more stable, less likely to get broken apart. Okay. So during a spontaneous change, free energy decreases and the stability of a system increases. And equilibrium, like we talked about in diffusion and osmosis, is a state of maximum stability. Things are evenly spread out everywhere in diffusion, right? We're not going to see any more change. That is as entropic as it can get, so it's going to stay relatively spread out. So a process is spontaneous can perform work only when it is moving towards equilibrium or moving from higher to lower energy states, okay? Or from less stable, higher, right, to more stable, a lower stance. Here we can see in this picture, the person up on the bouncy diving board has got higher free energy, but they are less stable and they have a capacity to do some work because of their height. But as they go down, as they dive down, once they get into the water, they're going to be at a more stable point, right? They're not going to fall once they're in the water, but they will have less free energy and less capacity to do work. They've given their energy off to the air, whatever. Diffusion, we can see there, when it's highly concentrated, it's at a higher free energy state. It is less stable. It doesn't want to stay that way, right? It wants to spread out. So it'll spread out, and that takes it to a lower free energy state, where it is more stable. And on the far right here, we see the chemical reaction where we're breaking down glucose, it looks like. And we're breaking that glucose down, which is a larger, more complex molecule, which is less stable than simpler molecules. It's got a lot of free energy in it. If we break it apart into its parts, we can get water and carbon dioxide out of it. Those individual molecules are more stable, and we can we'll release energy, right? We go from a higher to a lower state or from less stable to more stable state. That releases energy, and we harvest that energy through the process of cellular respiration to do the work we need to do. So that is energy and how it works. Going along with that, we have exergonic and endergonic reactions, which is very similar to what we talked about with catabolic and anabolic reactions. An exergonic reaction proceeds with the net release of free energy. So it exa, like out. So that's what we're talking about happens spontaneously. That's going from a high to a lower energy state, and we release that extra energy that we can use for work. Those are spontaneous. Endergonic are ones that you need to put in, put in some energy, so it'll absorb free energy from somewhere and use it to make, in the case of chemical reactions, more complex molecules. It's not spontaneous because it's going from a lower energy to a higher energy state, right? So you got to take some energy to push it up that hill to get to that higher energy state. So it's endergonic. This is the type of reaction that stores energy for us in molecules like glucose or ATP. Those are endergonic reactions when we're storing the energy. When we're releasing it, it's exergonic. Here we can see a nice diagram. This explains it better to me. You can see we've got on the top an exergonic reaction. And we can see right here we got the reactants. Here's the amount of free energy in this system and the reactants. If we can drop it to a lower free energy state, okay, if we lose energy, get simpler, break things down, that in that process, here's the energy state of that material afterwards, but that releases energy. That change in energy is negative G, right? Negative change in G. We've lost energy, it's gone off somewhere, and hopefully we can harness it to do work for a living system. Down here, we've got the opposite. We got smaller reactants. In order to build them up into something bigger, we're going to need to add energy, and that will raise the amount of energy in the system. So we have a positive G. Okay? So endergonic, we build up to get to a less stable but more energy stored state. That's positive G. Negative G, we lose energy, but we can drop down to a more stable state, and we can use that energy that was given off to do some work. So equilibrium and metabolism. So if we're in a closed system, we'll eventually reach equilibrium. So if we were doing diffusion in the room, when you guys were moving around in the room before, when we did diffusion, and I made you pretend to be molecules, packed you into the corner, you had a high concentration, and you moved out to the room where you were at a low concentration, right? In that motion, if we'd set up some sort of mechanism, we could have harnessed that energy to do some work when you were doing that motion through the room. We didn't. We could have. But eventually, we reached an equilibrium where we're evenly spread out in the room. And at that point, we weren't able to harness, we wouldn't have been able to harness the energy at all. And we would have been producing nothing, no work getting done. That would have been G is 0 at equilibrium. And that was a closed system, though. Question is life a closed system? So we'll come back to that. So the controlled release of energy is essential. Cells are not in equilibrium. We are open systems, experiencing a constant flow of materials and energy. A defining feature of life, guys, is the metabolism is never at equilibrium. If it's at equilibrium, we're totally entropic and we're dead. Okay? Cells are always using up oxygen and creating carbon dioxide in their bodies, for instance. That never reaches equilibrium. All right? A catabolic pathway in a cell releases free energy in a series of reactions. So catabolic, remember, breaking things down, exergonic. Why do you think it's important for a cell to release energy from molecules in a series of reactions as opposed to all at once? Hopefully you're thinking with me here. If you release it, all that energy was stored, say, in a molecule all at once, it's going to be much harder to harness it. It's kind of like if you take the amount of energy, we'll compare a firework to a fireplace. Okay, they're both combustion reactions. They can both release a lot of energy and heat. That firework's going to release it all at once, big old explosive reaction, and we can't do much work with it. The fire in the fireplace, we can control it, we can slow it down, and then we can harness that to do work like cook our food, heat our water, heat our houses, those kind of things. Another nice thing is we can keep adding to that fire slowly to maintain it for a longer period of time. Whereas that firework, it goes and it's hard to keep it going because it is all at once. This kind of shows that concept here again, where living systems are open systems. Here we can see you got water coming in at the top end. This would be the energy coming in from, say, the sun. Always having to add energy on this end if you want this thing to keep going, because it's always going to be leaving on the other. We're losing energy. But if we can do that, we can do work. We can have negative G there. We do it in a series of small steps as opposed to all at once, because we can get a lot more work done and we can harness it much more efficiently in these smaller steps. Each step along the way, we can harness some energy, we can harness some energy, we can harness the energy. We also lose some energy each step along the way, but we can make better use of it in a multi-step system, which is what we saw in that very first slide where we had product A to product D with multiple steps in the middle. That's part of why we see that in living systems. All right. In the past, you guys have learned about ATP, so right now we need to do a quick review of ATP and then add in some stuff you may not have realized about it. in light of our understanding of thermodynamics, energy, free energy exchange, exergonic, endergonic stuff that we learned about in the last little bit of notes. So ATP powers cellular work by coupling exergonic reactions to endergonic reactions. Okay, coupling like a train coupler, right? Where you got that little thing in the middle where the train comes and locks together so that it can pull the other cars. That's the coupler, and that's what's going to transfer the energy from the engine to the car, right? The coupler. So the ATP molecule is like that coupler. It's what's going to transfer the energy from the exergonic reaction, the engine, to the endergonic reaction, whatever it is we need to do or move. Okay, and the cell does three main kinds of work: chemical, transporting things, and mechanical work. And to do that work, we manage the energy resources by that energy coupling. The most common molecule to do that is ATP. There's some others we'll learn about, but ATP is the big one. Most energy coupling is mediated by ATP. You see in that last one. It's kind of like, I wish I could find this where it only went one direction, but we have a Newton's cradle here, right? As this ball falls, this is an exergonic reaction, right? It's going from a higher energy to a lower energy, less stable to more stable. It's going to give off energy. These middle balls are the coupling. This is like the ATP or the pathway of energy coupling if it's more than one. Sometimes that's the case. And it transfers its energy at the end to this last ball, and that triggers an endergonic reaction, right? This takes energy to move this thing up. So this one falls down, releasing energy. These transfer the energy and push this one up. So this would be that exergonic transferring energy to an endergonic reaction. Now in the process, we lose some energy, right? This one's losing it as sound and some little extra motions in there. But the same thing happens in life. We're going to lose energy in all these processes of transferring it. We can't be 100% efficient. Love to be, but we can't. All right, so let's take a look at the structure of adenosine triphosphate, which means an adenosine molecule with three phosphate groups stuck on the end of it. So here we can see we've got an adenine, a ribose, and here are our three phosphate groups. Okay, that's ATP. If we break off that last phosphorus group through hydrolysis, adding water, we wind up with adenosine diphosphate, two phosphoruses, and an extra inorganic phosphate group over here. This is breaking things down, right? That means it's catabolic, exergonic, releasing energy when we go to this simpler state. So you can see we got the energy over here. Now, if I were to reverse this and add that phosphorus back on to go up to ATP, that would be an endergonic reaction, right? We'd be storing that energy in this ATP molecule. And that's what happens when we charge up the ATP. I like to think of ATP and ADP as our charged battery and our uncharged battery. Okay, we can charge up our battery by adding some power. If you plug in your rechargeable battery into the wall, you can get power from the power plant. It'll charge up the battery. That's like putting this phosphorus on and going up, move uphill to that higher energy state, which is less stable. ATP is not a very stable molecule. It falls apart pretty easy, which is why it's a good energy coupler. And now we got a charged battery, and then we can use that battery, stick it in our phone to power what we want to do, or stick this in, the cell membrane to make it move if we're doing like exocytosis. And we can then drop down in energy levels, okay, drop down in free energy, break off that phosphorus, and release some energy. That's how it works. Okay. The bonds between the phosphate groups of ATP tail can be broken by hydrolysis. We saw it in the last one. That energy is released from ATP when that bond is broken. It doesn't come from the actual breaking the bond. It's because it drops to a lower energy state. We start up here where we've got the more complex molecule going to smaller molecules, less complex, entropy increasing. When we do that energy is released, and we got a negative G. And if we can harness that well, we can use it to do work in the body. So in the cell, the energy from exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. It can give its energy to something that needs it in order to go. Right? So overall, the coupled reactions are exergonic. We're going to lose some energy. Okay, so overall, it's exergonic. We got to take in more energy than we actually use. We lose a lot of it as heat and motion and things of that nature. So overall, we're exergonic. Okay. ATP drives those endergonic reactions by phosphorylation. In many cases, by transferring that phosphate group, that extra one that comes off, to some other molecule, which causes that molecule to change shape or do something. And that recipient molecule is now called a phosphorylated intermediate. It's that intermediate stage in between doing some work, and we add the phosphate group, it does something, and then the phosphate group comes off and it does something else. So let's take a look at this in terms of the energy. So here's just a simple reaction. Glutamic acid and ammonia, don't need to memorize this reaction. But putting these two things together, making it more complex, that's endergonic, right? Takes energy to do that, so we got a positive G, right? We got a plus 3.4, so it takes 3.4 kilocalories per mole for that reaction to happen. We need to add energy to make that happen. We can add that energy with ATP. Okay, glutamic acid, add in the ATP. There's that phosphorylated intermediate. We take the phosphorus off the ATP, add it on the glutamic acid, and we wind up with ADP, right? Those are intermediate stage, and then we can add these, bind these together. We wind up with a glutamine and ADP and phosphorus. We are now at a overall lower energy state. This is the real key for this whole thing down here to me. To go from these two combining to one molecule, we need to add in 3.4 kilocalories per mole of energy. But to go from ATP to ADP and phosphorus, that exergonic reaction actually gives off 7.3 kilocalories per mole. It's a negative G, like twice as much as it takes for the reaction to happen. So we lose like half the energy that was stored in that ATP, but we get the reaction to happen that we need to happen for life to go. So we couple this exergonic reaction with an endergonic reaction. This powers that, but we do lose some energy, right? Overall, the energy drops. We lose energy somewhere. Okay. And we can see on this slide just some of the other ways that we use ATP for transporting molecules, for some mechanical work within the cell. We can do that using ATP. So this kind of shows how ATP is the coupler between two reactions. Over here, off the screen, is some reaction that's giving off energy. Okay, maybe it's cellular respiration, right? It's giving off energy. We use that energy to charge up the ADP into ATP. This is an endergonic reaction over here, right? We got to put energy in to make the molecule more complex to store that energy. Over here, if we hydrolyze ATP back down to its simpler form, we drop its free energy. We got a negative free energy over here, exergonic, and that energy that's given off can be used to do some reaction over here. So we got energy going in from some reaction over here, makes the ATP. When we split it, the energy goes out to do something else. So ATP is that midpoint. It's that rechargeable battery in the middle, constantly spinning around between whatever is producing our main source of energy over here and wherever you're using that energy for over here. All right, and that does it for our review of just energy concepts. In the next set of notes, guys, we're going to take a look at specifically enzyme structures and catalysis. In other words, the properties of enzymes and how they work.